Hello everyone, welcome back to my YouTube channel. So today's video it's all about the respiration and reproduction in bacteria. Bacteria breathe using oxygen, it's called aerobic, and without oxygen is anaerobic. So there are types of bacteria and depends on the respiration. Okay, oxygen absent or present. Okay, so obligate aerobic bacteria must have oxygen to survive. Obligate aerobes are bacteria that must have oxygen to survive and grow. They depend entirely on aerobic respiration, which uses oxygen as the final electron accepted in the electron transport chains to produce the energy ATP. Example of obligate aerobic bacteria, that is Mycobacterium tuberculosis, which is caused the TB. Mycobacterium tuberculosis, the charming microbes behind tuberculosis, as we know, is an obligate aerobe, meaning it needs oxygen to survive. Right? It thrives in the high oxygen environment of the lungs, especially the upper lobes, where oxygen concentration is highest. Second one, obligate anaerobic bacteria. So die in the presence of oxygen. It is the totally opposite. Here it is the full oxygen concentration, it is the zero oxygen concentration it's required. Right. So the obligate anaerobes are bacteria that cannot survive in the presence of oxygen. So oxygen is actually toxic to them because they lack the necessary enzyme. Okay, like catalase and superoxidase dismutase. Yes. So to neutralize harmful oxygen byproduct like superoxide radicals and peroxide. So when oxygen stop, these guys just peace out permanently. Right? So the example of Clostridium tetani causes tetanus. Clostridium tetani, as we know, the bacteria responsible for tetanus is an obligate anaerobos, meaning it cannot survive in the presence of oxygen. Right? It thrives in deep oxygen poor wound like puncture wound from nail or splinters, where there is minimal oxygen and plenty of nutrient to sporulate and produce the tetanus parsimine toxin. This is a neurotoxin that causes those painful muscles contraction in tetanus. Third, facultative anaerobic bacteria can use oxygen if it's there but can live without it too. Okay. So facultative anaerobic bacteria that ultimate survivor, they are flexible, yes, okay, and can adapt to different environment if their oxygen is present or not. When oxygen is available, they use aerobic respiration, which generates the more energy. While when oxygen is not available, they switch to anaerobic respiration or fermentation. Ability, okay, so toggle between oxygen rich and oxygen poor condition and gives them serious advantage in varied environment. Example, E. coli in human intestine. So E. coli is a classical example of facultative anaerobic bacteria, okay, where it lives in the human intestine where oxygen levels are low or variable, and it adapts beautifully. When oxygen available, like certain parts of the gut or outside the body, it performs aerobic respiration for efficient energy production. So now when oxygen is absent, like deep in the colon, in human colon, okay, it switches to anaerobic respiration or fermentation. Now, fourth one is aerotolerant anaerobes. Don't use oxygen but are not harmed by it. So what do you mean? Aerotolerant anaerobes do not use oxygen for the energy production. They rely on fermentation. Okay, are not harmed by the presence of oxygen, but they can survive just fine around it. They have enzyme like SOD, that is superoxide dismutase, that help them detoxify reactive oxygen species, even though they don't actually use oxygen themselves. Now, fifth one is microaerophilus. Okay, wait. Here is some of the example of aerotolerant anaerobes. These bacteria are commonly found in yogurt, cheese, pickles, human gut, and vaginal microbiota. Now, microaerophiles need oxygen but only a small amount. Okay, a small amount. Need oxygen to survive. High concentration of oxygen, like in normal air, are toxic to them. They typically thrive in environment with 2 to 10% of oxygen compared to minus 21% in the atmosphere. So example of microaerophilus Helicobacter pylori, which lives in the human stomach, which has low oxygen, associated with peptic ulcer and gastric cancer. So this is respiration bacteria. There are five types of respiration in bacteria. Now come to the reproduction in bacteria. Bacteria mainly reproduce asexually. Asexual reproduction is the process by which a single organism produces offspring without involving gametes, sac cells. Okay, so the offspring which produce there are usually genetically identical to the parents. And the most is a binary fission. Okay, mainly reproduce asexually using binary fission. So now the single bacterial cells divides into two identical daughter cells. Now here we take the one of the example rapid process under ideal condition. One cell become 2:32 equals to approx 4.3 billion cells in 10 to 12 hours. Okay. So here bacteria reproduce by binary fission, meaning one cell divide into two cells, and this process can happen as quickly as every 20 minutes under ideal condition. What is ideal condition? That is like optimal temperature, nutrient and moisture available for growing bacteria. Now let's go through the step by step to understand how bacterial cell population, okay, multiply and how we get to 2 to 32 equals to 4.3 billion cells. So first, what is generation in bacteria? Binary fission. One bacterial cell divides into two identical daughter cells. This process called one generation. Now, after one generation, one cells become two cells. After two, two to four, four to eight, and so on. Now, how long is one generation under ideal condition? Like nutrient availability is properly nutrients available in the medium, temperature, proper temperature, pH. Many bacteria like E. coli divide every 20 minutes. If one generation, that means one generation required 20 minutes. So how many generation in 1 hour? 1 hour, that is 60 minutes. So the 3 generation per hour. Right. And we have to calculate the 10 to 12 hours. So how many generation in 10 to 12 hours? 3 generation into 10, that is 30 generation. And 3 generation into 12, that is 36 generation. Right. So 10, 12 hours you get between 30 to 36 generation. Now final. Exponential growth formula where the bacteria are growing in well manner with optimal growth factor, nutrient, temperature, and pH. Each generation doubles the number of bacteria, so the total number of cell after n generation, number of cell 2 n to 1, n. Okay. So here you just put the 2 is to 32, right? And answer is 429467296 is equals to 4.3 billion cells. Now come to the types of asexual reproduction. First, binary fission. Okay. Second, budding. Third, spore formation. So binary fission most common, and one cell split into two. Budding, a small outgrowth forms and become new cells. Spore formation, in harsh conditions, some bacteria form tough spores. So first we understand the binary fission. Okay. This is the most common method of asexual reproduction in bacteria. It is simple and efficient process where parental cell bacterial DNA, here it is the parental bacterial DNA replicates, cell grow in size, the cell membrane pinches inwards, okay, here it pinches inwards. And then the cell split into two genetically identically daughter cells. So this method also allows bacteria to reproduce rapidly, especially under favorable conditions. Now, second type of asexual reproduction in bacteria, that is budding. A small outgrowth forms and becomes a new cells. Okay, here you can see in this picture, right? So budding is also is a type of asexual reproduction where new cells, okay, develop a small outgrowth. This is the outgrowth on the parent cell. Yeah, you can say the bud also. Bud. Right. This bud enlarge, develop its own DNA. This is own DNA, and eventually it gets to become a new independent cells. Although rare in bacteria, some species like planktomycetes to reproduce by budding. Okay, so example of planktomycetes. Okay, so it is also see budding like this. Now third, asexual reproduction, spore formation. In harsh conditions, some bacteria form tough spores. Some bacteria, especially gram-positive, gram-positive types like Bacillus and Clostridium can form endosperm, highly resistant dormant structure formed inside the bacterial cell. Here it is to see internal spore formation inside the cell and outer side of the spore formation. Okay. So why do they form spores? What is the main purpose of forming the spores? For survival. Okay, spore formation for survival, not a reproduction. When bacteria face unfavorable conditions like high heat, dryness, UV radiation, lack of nutrients, chemical exposure, that time they have to produce the spore. Okay, spore, that is they form a spore to protect the DNA and essential compounds. Okay, so this is like a one type of chemical structure. I think biomolecules. Okay, they cover up the DNA and other essential compounds in the cell. Now these spores can stay inactive stage for years, and when condition improves, that condition are favorable in condition, okay, they can germinate back into active and growing bacterial cells. Okay, so these are the three types of sexual reproduction in bacteria. Now come to the sexual reproduction and genetical recombination in bacteria. So sexual, it's not a sexually in the usual sense, but involves DNA transfer to create variation. Okay, so first there are three methods. Yes, one transformation, second conjugation, and third transduction. So now first we understand the transformation. Okay, so who discovered the transformation process? Okay, scientist Frederick Griffith in 1928. So organism use Staphylococcus pneumoniae, a bacteria that cause the pneumonia. Right. Now, types of bacterial strain used by Griffith, they were used two types of bacterial strain. One is the S strain, smooth, and second one is the R strain, it's tough. So smooth strain have a polysaccharide capsule, okay, has a polysaccharide capsule, and virulent causes the disease. It's toxic compound which is produced by the S strain, and they cause the disease. Okay. R strain, that is rough, no capsule, no virulent, okay, that is harmless. Griffith experiment, so treatment, live as strain injected into the mouse, so mouse is died because it has the virulent toxic compound. Now second, live R strain, okay, injected into mice and mouse is live because non-virulent, there is no any harmless compound present in bacterial cell. Now third, heat killed S strain. Heat killed S strain, but S strain is heat killed and they're dead. Okay, so the mouse is live. Okay, harmless, no any side effect. Okay, now heat killed S strain plus live S strain. Both are injected, mouse dies. An unexpected result. Okay, so the after injecting a mix of heat killed S strain and live S strain, the mouse died. And live S strain bacteria were recovered from the dead mouse. Okay, live S strain bacteria were recovered from the dead mouse. S strain is already killed. But however, after the experiment, live S strain bacteria were recovered from the dead mouse. Okay, so here it is. What is conclusion behind this process? Okay, so some transferring principle from the dead S strain into the live R strain. Okay, transforming principle. Let's say that the dead S strain entered the live R strain. This transformed non-virulent R strain into the virulent S strain. The transformation R strain acquires genetic traits like capsule formation from the S strain. Okay. So now death of donor bacteria S strain. Okay, donor bacteria S strain. So it is the DNA. Okay, they did and DNA into the environment and the uptake by recipient bacteria, that is S strain, and S strain become R strain, and R strain become S strain, and they produce the virulence. Okay, so how we conclude that? Okay, here release DNA into the environment. Okay, by the S strain, and R strain uptake this DNA. Okay, and this DNA produce the virulent. Okay, and this R strain convert into the S strain. Okay, and cause the disease. Okay. Now, one another fact of this experiment that later in 1994, Avery, McCaldy, and McCarthy identified that the transforming principle was DNA, proving that DNA is a genetic material. Yes. Okay. Okay. So second sexual transformation in bacteria, that is the conjugation. So conjugation, we discovered the conjugation process. Joshua Lederberg, Edward Tatum in 1946. Organism used it is the Escherichia coli and first demonstration of genetic exchange between bacteria. What is conjugation? It is direct transfer of DNA from one bacteria. It is the one bacteria, okay, it is the donor, to another bacteria. Okay, so this is the recipient, second bacteria, a physical connection called the pilus. It is the pilus, or you can say the conjugation bridge. So some of the key terms we have to know about this. F plus cell, that is the donor cell, contain the F plasmid or a fertility factor. F minus cell, that is the recipient cell or lacks the F plasmid. Now F plasmid, if F plasmid which is present in donor, only donor, okay, on donor cells. So this is a small circular DNA that contains genes for pilus formation and DNA transfer. Now sex pilus, that is this is the, okay, a protein tube or bridge that connects F plus cells to F minus cells and HFR cells. Okay, HFR cells. So some of the variation of cell having the F plus plasmid or HFR. Okay. So variation of A plus where the F, AF plasmid is integrated into the main chromosome or main genetic material of the DNA and can transfer chromosomal genes to the two also. Okay, chromosomal gene also transferred by the pillars. Now we understand the step-by-step procedure of conjugation. So first initiation or recognition and contact. So first initiation, recognition, contact, right? So it is the first, the A plus donor cell recognize and F plus minus recipient cell. The F plus cell extend as a sex pillars, okay, looks like this, and which attaches to the surface of F minus cell. The pillars retracts and bridge the two cells into contact. Second, formation of conjugation bridge. Next is a formation of conjugation bridge, mating bridge, protein channel formed between the two cells. Mating bridge, you can also say the protein channel. Okay. The bridge allows direct transfer of DNA. Now third, replication. Okay, third, replication and transfer of F plasmid. So in the F plus cells, okay, this one, the plasmid is nicked at the origin of transfer, ODT. A single strand of the plasmid DNA is transferred to the F minus cell through the conjugation tubes. Its transfer occurs both cells synthesize complementary DNA strand, yes, and restoring double standard F plasmid in both cells. Now this one is also F plus cells. Now completion, last step. The F minus cell now become F plus, having received the entire F plasmid. Both bacteria are now capable of initiating conjugation with other F minus cells. So now here it is, why is conjugation importance in bacterial cells? Okay. So major method of horizontal gene transfer among bacteria. This is horizontal gene transfer. Spread beneficial gene, especially they spread the beneficial gene. If this bacteria having the penicillin resistance genes, they can transfer to the another bacteria. Okay, so antibiotic resistant genes, virulence factor, metabolic capability. So next, third type of sexual transformation in bacteria, that is transduction. Okay, so transduction is easy process. Okay, you can understand. I think all of the process are very easy. Okay, so first, any gene from the bacterial genome can be transferred. Right. So first phase infection, phase, that is the bacteria phase, that is bacteria. Okay, there is also called virus. Those virus infected into the bacteria, this is called the bacteriophage. So lytic bacteriophage infects donor bacterium. The phage inject its viral DNA. It is the viral DNA, okay, into the bacterial cell. So the phase and attachment first the cell surface and the bacterial DNA break. Okay, bacterial DNA. Phage DNA inserted into the bacterial cell. Now bacterial DNA breakdown. Here is the okay. The phage takes control of the host and the bacterial chromosome is broken into fragments. Okay. Now mistaken packaging. Sometimes during assembly of new phages particle, bacterial DNA fragment instead of viral DNA is accidentally packaged into phage head. Okay. So this creates a defective transducing phase. Sometimes bacterial gene are also involved into the viral gene, okay, phase particle. Okay. So it is a recombination, recipient gain specific traits, unlike the virulence or resistance, right? Now, infection of recipient cell. Okay. So the transducing phase infect a new bacterium cell. It inject bacterial DNA, not viral DNA. Okay. So now what will happen? Now the phase integrated but temporary phase infection incurred exclusion increases exercise. Okay. So here it is the phase is phase DNA contain the bacterial DNA and bacterial DNA which transfer to another cell of bacteria. Now DNA recombination. Okay. So the foreign bacterial DNA may recombine with the recipient genome. This leads to genetic variation. Okay. So this result, recipient bacteria can new genetic traits like metabolic changes or resistance, or you hybrid DNA also formed by the uh, transduction procedure. Okay, so this is the all about today's video. I hope you find this video more informative and valuable. Thank you for watching this video and don't forget to subscribe to my YouTube channel. And next lecture we will be discussing about some common bacteria. Thank you for watching.